someone is voting, it'll start to go up and then asymptotically.
All right, I will start the webinar right now.
Excellent, thank you.
Stop the count. Oh dear.
Go on mute, Adrian.
So there are still more participants dialed in. We are already at more than 170. Let's wait for another few seconds maybe and then start. All right, very warm welcome, everyone. I'm truly excited to moderate the kickoff meeting of the Target 2035 meeting series. My name is Anke Müller-Farnow. I used to be head of lead discovery for Bayer AG for more than a decade. In my current position, I'm now senior scientific advisor at the newly formed Nuvisan Innovation Campus Berlin. And since July this year, I have the great honor to be chair of the SGC Board of Directors. Let me start with two technical notes while maybe a few more people are dialing in. The first one is this webinar is recorded and it will be available via the Target 2035 homepage. The second more important technical note is there is a Q&A function here in the meeting. So if you go to the bottom line, it says on my screen at least F&A, which means question and answers. So if you want to ask a question, please just type it in and we'll try to get to that question either as part of the panel discussion or maybe afterwards via the homepage again. So now let's start. Imagine what we could do in academia and in drug discovery if we had a chemical probe for each and every human protein. I think that would be just fantastic. And you will hear more about that from Cheryl, Adrian, and Aled, and of course also during the panel discussion. I think it's a really exciting idea to achieve this kind of ambitious goal. Before we start with the agenda, I want to thank the organizer. Thanks to Claudia, to Christina, and to Ushi, who did all the organization, and of course also to the program committee, who all worked together very hard to make this, I think, a very appealing program. At least we have now more than 200 participants already dialing in, and I think there's also a lot of interest for the next sessions. So our first speaker opening up the series is Cheryl Aerosmith. She is a chief scientist of the Toronto node of the Structural Genomics Consortium, and she leads the SGC's program to develop chemical probes for chromatin regulators. Cheryl is especially well known for her contributions to structural and chemical biology of chromatin and epigenetic regulatory factors. Cheryl, the floor is yours.
Good afternoon, good evening, and good morning, everyone. I am sharing, oh, sorry. I'm going to share my screen here. All right.
Excellent.
So to introduce you to Target 2035 is the purpose of this webinar series. And I'm glad so many of you could join. So we, in the leadership of this initiative, want to let everyone know in this kickoff series, webinar series, the why, what, and how of this initiative. So I will just give a very brief overview of what you're going to hear in this upcoming webinar series and how you can participate. So what is Target 2035? It's an initiative to develop pharmacological modulators for most or ideally all human proteins by the year 2035. It's an international federation of biomedical scientists from industry, small biotech, and academia and not-for-profits. Basically, everybody who can contribute is welcome too. We want to develop, apply, and share new technologies in order to get there, because it's a very ambitious, some may say impossible at the moment, initiative, but we think it is possible with the right organization and global participation. And this is going to take open science to share our knowledge and expertise and develop these new technologies. So the concept came about through a meeting of scientists shown here hosted by Bayer when Anke was there, and in Berlin in 2018. And this was key thought leaders in the field, members of multiple pharmaceutical companies and academics. And we sort of came up with this concept that you're going to hear about over the next few days of trying to identify a pharmacological modulator of most human proteins to better understand human biology and disease and facilitate drug discovery. So that leads me to why. You're going to hear today talks by Alan Edwards on the academic perspective and basic science perspective of why we need to do this. Adrian Carter is going to present the pharmaceutical industry perspective. And then we're going to have a panel discussion with members from many different perspectives, industry, academia, scientific publishing, etc. So in a nutshell, why we're doing this is because drug discovery is slow and expensive, and most new drugs actually fail in the clinic when they're in the clinic for the first time. And this suggests we as a community have an insufficient understanding of human biology and disease and how to target it. And 20 years after the human genome was sequenced, there's still, most human proteins are still undercharacterized, and there are not sufficient reagents to probe their role in disease and understanding of human biology. Because pharmacological modulators are among the most useful tools for biological and drug discovery, we think that as a community, we can contribute to making such pharmacological tools to remedy this situation. So what is involved in Target 2035? We're going to hear about that over November 11th, tomorrow. We're going to hear examples and learnings from projects focusing on the kinome, epigenetics, GPCRs, and also perspectives on opening up new target classes that are not typically considered druggable. So one concept that I want to point out, and you're going to hear more about it in the coming talks, is key features that are required for a useful public domain pharmacological tool. The tool should be unencumbered with no restrictions on use, so scientists should be able to use it as they wish for research. It should bind directly to the target of interest and modulate it. It should be cell permeable, at least for, at a minimum, able to modulate the target within a cellular assay, and if it has properties for in vivo studies, that's even better. And it needs to be well characterized in terms of its selectivity especially as related to similar proteins in the proteome. The corollary is that it should be non-toxic due to off-target activities, and generally not a highly reactive or promiscuous agent. And some examples of these types of tools are shown here. A chemical probe is ideally what we'd like to each target, but also biological probes to extracellular and cell surface proteins would be certainly useful. Chemogenomics libraries is another concept you're going to hear about in the coming talks. And then this concept of protein handles in the age of PROTACs and proximity pharmacology. A protein handle is a small molecule that binds to one protein such that you could create a chimera or a molecular glue that would bring it into proximity of another protein to modulate it. And here are some papers that describe high-level concepts for these types of chemical modulators. And then longer term, how to achieve target 35. On November 12th, we're going to hear about ideas to accelerate chemistry and chemical biology, because we'll need to make these discoveries in a faster, more efficient manner. We're going to get there by 2035. And we have a special focus on Asia webinar on November 16th. It's at a time slot that's more convenient for the Asian audience. Okay, and then what we want to do after this week is to have a monthly series. This is going to be the second Monday of each month. The dates are shown here with a more focused discussion. It's still going to be a 2-hour window. And December 14th is a targeted protein degradation and proximity pharmacology focus. January 11 is computational and artificial intelligence assisted methods for ligand discovery and optimization. In February, we're going to talk about chemoproteomics and proteome-wide methods for characterizing modulators of proteins, and so on. So how can you contribute? We want this to be a global initiative. Here's the contact email if you would like to somehow get involved. So what can you do to get involved? Think about how your research can contribute and participate in this group of organizations working towards these goals. Some of the organizations I have listed here. If you've already made chemical probes or tools, donate them, in quotes, donate, make them available, maybe make a small amount available, and we're establishing infrastructure to be able to do that and make it available to the community. Collaborate with your colleagues and colleagues you may meet at this series to make new chemical tools and novel proteins. Invent and share new technologies. This is going to be so important to getting there by 2035 to accelerate drug discovery and evaluation of chemical modulators. And join us if you have an idea and you want to organize one of our focused webinar series on a particular topic. Get in touch, and we'd be happy to facilitate a program that would focus on a specific topic. So I'm going to end with an ad for the first focused meeting after this initial kickoff webinar series, and this is hosted by Ingo Hartung. And you can see some of the speakers here, and that will be on December 14th. So for the next 3 days and next Monday in Asia, we have a great program for you. And I'm really excited to hear our speakers and get all of you involved in the program. Thank you.
Thank you very much, Cheryl, for this introduction. There was already one question that you might like to answer briefly, and that is, does Target 2035 include biologics, secreted extracellular proteins, or only small molecules?
I think biologics, yes, for that can modulate cellular systems. So those that can modulate cell surface proteins and cell surface receptors, yes.
Okay, thank you very much, Cheryl. With that, I'd like to introduce our next speaker, although I'm sure he does not really need an introduction. It's Elliot Edwards, who is the founding and current CEO of the Structural Genomics Consortium. He's a professor at the University of Toronto and adjunct professor at McGill University. The Structural Genomics Consortium, or SGC, has been sharing its science freely since 2003 already and is widely acknowledged as the pioneer in open science. And Elliot is really a driving force here, making sure that as much research is freely available as possible. Elliot, looking forward to your presentation.
Thank you. And does this work? There we are. And so I'm happy today to sort of give what I believe is sort of the background as to why one even would want to do this and why now. If you look from 30,000 feet or 9,144 meters, we around the world every year, we spend about $300 billion in biomedical research, half the private and half the public sector. And over the course of the last 50 years, we've made tremendous advances, and you can point to some of the enormous improvements in human health that have occurred from our research endeavors. But at the same time, for many diseases, we still don't have a therapeutic strategy. We don't understand the mechanism. Parkinson lived in the early 1800s when he described that disease. We still don't have a mechanistic understanding of why people get Parkinson's and how to treat it. So there's huge gaps. And the reason what I'd like to get across today is some reasons for those gaps. And the basic reason why we are struggling to understand disease and develop therapeutics is, as Cheryl said, we don't really have a good understanding of human biology. And that is the common problem that faces both industry and academia. And our common goal is, how do we get to a complete understanding of human biology faster? It'll happen. It's not a matter of if, it's a matter of when. And so our thought should be about speed and efficiency as opposed to anything else. And so I like to use the analysis that Adrian is going to show later to sort of give a framework for how to think about this problem. And in this analysis, we take advantage that the human genome is bounded. And so it's one of the few areas of science, capital S, in which you can map activity onto a bounded scientific problem. If you were to think about materials science, there's an infinite number of molecules you can make. So in an infinite number of, there are only 20,000 genes. So we can say this, how does science relate to those 20,000? And we just took the protein kinases here because of the practical simplicity of measuring. And so on this axis here are the 518 kinase genes in the human genome. On this axis here is the papers. So if you see the kinase name in the title, the abstract, or keywords. This is now a very common way of representing scientific activity. And you can see prior to the human sequencing of the human genome, much of the activity was on the genes that had been discovered at that time and that were popular. Now, almost everything is a power law. So don't, the power law is a common mathematical, outcome of plotting something like this. So don't pay much attention to that. But what you can do is you can do the exact same search, SARC kinase, and then you add and 2017. So now you're going to look 17 years later, where was the world publishing? And I'm going to keep the kinases in the same order, ranked on the popularometer by pre-2000. And what you can see is that with some exceptions, that's BRAF, those are the Parkinson's kinases, most of the scientific activity on the planet, most of the funding, most of the people are still working on the kinases that were the hot ones prior to the sequencing of the human genome. And this is explicable to anybody who's written a grant or tried to get papers published. It's so much easier to work where people know about stuff, and it's really hard to develop hypotheses about something that is unknown. Now, so our system of funding and writing grants and publication apps kind of resists change, right? And if you were to ask someone who's not a scientist and say, if you had $1, where would you spend it? Would you spend it to be the 10,000th dollar here or the first dollar down here? Where would you get society's biggest return on investment? They would probably say, an economist would say, work down here. but our system makes us work up here. So that's one problem in our system. The second problem in our system is science is not a global endeavor. Canada does not fund science to make the world better. Canada funds science for our economic development. And as a result, we fund the best science in Canada. And so if you take the exact same search string that you used here, SARC plus 2017 plus Canada, out of this global picture, you'll only get the Canadian papers. And you'll find out that it is actually just a small subset, you know, a few exceptions, but by most and large, Canadian science is a representation of the global science. You know, I sometimes say if every Canadian was hit by a bus tomorrow, it would not actually affect global science advance much because we're doing much as what the world is doing. And for those, you know, we always talk in our fields about getting scooped. And another view of getting scooped is you're redundant because someone else is doing it, right? So I think there's a good sort of philosophical argument and economic argument to work down here. Our system doesn't do it, nor does our national way of doing it. And if you don't, and it's not just Canada, if you do it by university, if you do it by funder, or if you do it by country, nowhere in the world, the pattern is the same. So the takeaways, I would argue, from a pure how do we understand life, is to understand human biology and parenthetically to prevent diseases, we need to rebalance, I think, the intensity of where we fund research and re-emphasize a little bit towards the unknown, the proteins and the genes that no one works on. But as all of us in academia know, it's incredibly hard to shift one's research for proteins for which there is little known. You can't write a grant, and few reagents because it's going to take you forever to get the tools to do the experiment. The other thing we need to do is to re-emphasize that science should be a global endeavor. So in addition, of course, to promoting national interests, which is how the governments look at it now. So, you know, I think the talking points to our government should invest in science and prosperity will come, not invest in science to create prosperity, because that starts to get a lot of perverse incentives about funding national interests. So if you want to get a government to fund your science, say, hey, another country's doing it, we'll get left behind if they don't, if we don't fund it. And that's the common way to use it. But that means we're all funding the same thing. So, so how do you encourage, though, the community then? We're faced with quite a conundrum, right? We have a granting system that doesn't allow it. We have governments that fund the same thing over and over again. It's not really governments, it's actually us, the peer reviewers, but we fund the same things over and over again. And so there was a lesson from history that we noticed about 10 to 15 years ago, was that if you put chemical probes out there, they have a way of moving people. So I first heard it from Francis Collins. I don't know if he was the one that came up with it. It's hard to herd cats, but you can if you move their food. And so here's a similar graph, and these are publications on the 48 human nuclear receptors. And this is really work from Tim Wilson and his colleagues at GlaxoSmithKline. We're kind of pioneers in this. And so prior to the human genome and the sequencing of these genes by companies, you saw that a few nuclear receptors garnered most of the attention. But if you look on 2009, there was a downward shift in at least some of these receptors. And we looked at whether they were linked to disease, we looked at availability of antibodies, we looked at all sorts of parameters. And there was only one thing that we found that connected all these. For these and only these, industry had made a chemical probe available. And if you align the gene family by availability of probe, you see something kind of striking, right? It turns out that you could have prioritized these nuclear receptors by many different ways. Links to disease, as I said before, availability of mouse knockout, you know, genetic understanding, but no, chemical probe available, that's what garners the most people. Because as you all know, it's a great experiment to do. You know, you have a reversible, titratable molecule that can be added to any system to study the effects of activating or inhibiting it. It's a powerful reagent. And I would argue the most powerful reagent, if at least you look at the evidence from nuclear receptors. If you look down here, there's HNF4, that's mutated in mature onset diabetes of the young. It's not studied. there's no probe, right? So the idea was that maybe if we create new probes to proteins that aren't studied, it may act as a magnet to shift people from what they're studying to new areas. And so we started a pilot project in 2008, and the concept was, well, let's focus on an exciting area of biology, but maybe that was worth a little more attention. So we chose the protein families that modulate epigenetic signaling, so methyltransferases and the readers and writers of epigenetic marks. We wanted to clearly define a chemical probe, as it were, a target product profile, because probes are only as good as their quality. So you really needed to establish quality criteria in advance. You wanted to make sure that you didn't declare victory like on the aircraft carrier. And so we wanted to submit the ideas to an independent assessment before we made them available. We wanted to make sure, as Cheryl said, the probes were openly available. There was some evidence from the nuclear receptor studies that the probes that were made openly available were used more than the probes that were subject to restriction on use. And finally, to collaborate with industry, because at the time, and I think still today, most of the expertise to do this kind of pharmacology and medicinal chemistry lies within industry, and specifically within pharma. So the first thing was really important, and this is what Cheryl was speaking about. We wanted to get the community together to find what would a useful chemical probe be. It's not a drug, remember, it's a tool compound that enables you to study the biology of the target. And to do that, you need to have sufficient potency, you have sufficient selectivity, you have to make sure that it doesn't, selectivity within the family and outside of the family. It has to work inside cells and prove that the mechanism of action that you see is working, and of course, some other characteristics. And over the course of the last decade, the SGC team and their collaborators in industry have contributed a lot of probes now to the public domain, and each of these meets these criteria. As one example, the bromodomain family. Bromodomains are 57 odd proteins, domains, in the human genome. They bind acetylysines, and together you can see a lot of pharmaceutical companies have contributed molecules to the public good as part of this project, and we're very appreciative of that. And so the impact from the probe so far, if you'd look backwards, we've produced 100. They're not easy to make, right? Because you have to be very attentive to their quality. We've distributed more than 30,000 samples. They've been cited now in over 15,000 papers. There are literally hundreds of drug discovery programs in preclinical development where the hypothesis came from the use of the probe, and 40 clinical trials ongoing. And so, you are seeing a shift of research as more publications start to emerge on the proteins for which probes have been made available. So I think we're seeing early glimpses of successes, but it's 100 and there's 20,000 genes. How are we going to scale that? I think we can't do it simply linearly. We have to think of a more inclusive way of doing that. And the probes, I should say, just for the academics out there, they do generate cool science by others. So here are some papers. This is using the G9A probe that was made by Jan Jin and his colleagues at UNC. That's been used by others to make protein disease links. This one is a methyltransferase made in Toronto that in Oxford, a clinician scientist used to try and increase the expression of a disease-related gene. These are WDRs, a very cool protein class that are protein interaction modules. And this is a collaboration with between Toronto and Ontario Cancer Institute that developed that molecule. And they go on and on and on. And there are literally hundreds and hundreds of these papers now that use the probes to generate disease and biology links. As Cheryl said, there's other strategies. There's some protein families where it may be difficult to get a beautifully selective probe. But what Tim Wilson and David Drury are doing are using that to their advantage. And for the protein kinases, the concept is that we generate rather selective molecules that for each molecule, for example, will hit a number of kinases. And if you imagine in your head an overlapping Venn diagram where 5 + 5 + 5, you can get a limited number of compounds to cover many kinases, and then you can disentangle the results later once you get the hit. Now, Tim will be talking about this later, but currently, thanks to contributions again from industry, we have characterized 188 compounds that blanket 212 kinases with a minimum potency. And on the kinase tree, that's what it looks like. And the intention is to grow this set so the entire kinome is covered with a relatively, let's say, 500 to 800 compounds. And thus, with that one screen, you'll be able to ask the question, is there a human kinase that's involved in my biological effect? And what's super important about these, it's not the actual compound that's the value, it's the data behind the compound. These have been profiled extensively. And so if you get a hit with one of these compounds, you can look up all the things that are known about that compound. That's super duper important. And of course, there are papers coming from the use of that library that's been distributed to 30 people, 30 groups all around the world. And I think 75 or 80 papers have come from the use of that compound set that is being made openly available. What Cheryl said is we're now starting to get probes from the community that I think people are buying into the concept that high quality probes are of value, and that if they're peer reviewed, as it were, or rigorously reviewed, they'll have more utility. And so what the pharmaceutical companies are doing, and this was spearheaded by Anka, who's there, and Mark Bunnage, who's now at Vertex, and now has been uptaken by many of the pharmaceutical companies. They're starting to contribute molecules to the public domain so that they can be used by our community to do science. But it's not only the molecules, it's the data behind them. An incredible amount of characterization of these molecules that has occurred that have not been in the public domain, or at least collated in the public domain. And these companies are doing a lot of work going back into their databases, coming to an independent committee said, does this meet the criteria? And arguing that it does. And most do. 77 have been improved. And now these are being distributed by the lab, and 6,000 have been sent to 24 countries. And papers are starting to come from this. Here's just one example. I think this was a molecule contributed by Bayer that was used by clinician scientists in Toronto. And Cheryl was involved in this paper to look at some oncology things. So again, the advantage of having the molecule and the data available is that the scientists can do these kinds of experiments. And I think it's a tremendously exciting outcome. And hopefully we can get far more data and far more probes, as Cheryl mentioned before. But of course, probes are only as useful as their quality. And because it's a word that anyone can ascribe to their molecule, sometimes probes are used that should not be probes, as it were. The data aren't available, they're non-selective, and the biological result you see is actually not due to the presumed target. And a few years ago, we started an effort that the community could start to think about how to qualify, or how to sort of give their expert opinion on various molecules that are being discovered and that have been used in the public domain. And we just recently got funding from Wellcome to support this effort for 5 years, and Paul will talk about it later. You can ask him in the session. But I think this is critically important because, unfortunately, papers continue to get published with molecules that don't meet the criteria, that actually aren't useful molecules to uncover biology, and that's disappointing. It's a function of our review system, and it takes a long time to fix. So ideally, what will happen is this chemical probes portal will have expert opinion on the various molecules that are being used in the community, and we're delighted that Wellcome has decided to support it and give it robust. We've been trickling it along, but now with the funding that Wellcome has, I think that we can really turn this into a good community contribution. So as I want to just my last two slides, reiterate what Cheryl said. Looking forward, it's not achievable without some contributions from others. I mean, we've spent a considerable time and effort making 100 probes, and obviously that won't scale. So we're going to need many new experimental technologies. Selectivity at a genome scale is certainly something that'll be incredibly useful, and useful to also in toxicology studies. We need to explore a spectrum of modalities, macrocycles, proteins, peptides, nucleic acid, who knows, lipids, sugars, small molecules. You know, we should keep an open mind. We need massive improvement and then adoption of computational methods. The computational power far exceeds the data available, so we need, I think, to think about how to do that. And finally, global collaboration. We can't have each country or each institution going off piece separately in their own little silos, because this is a global problem, and too much money is already wasted on redundant science. So get involved, and this is the last slide. Again, what Cheryl was saying was that's the website. And if you want to specifically know who's sort of looking after each area in the general project, of course, Cheryl's leading it. The computational methods, Christina's already invoked a public-private discussion on how to improve those methods, and that's ongoing. That's in collaboration with Alex Hillish, who heads up computational chemistry at Bayer, and Pat Riley from Google. You want to contribute a probe for consideration to that donated probes, just make the attention Claudia. And if you're interested in helping out the chemical probe portal, Suzanne is the point person on that. So that's it for me. Thank you very much. And over to Adrian.
Thank you very much, Aled. I hope you can all hear me. I will try and share my screen now.
Maybe while you try to share your screen, a few words to you, Adrian. So Adrian Kahler is Vice President and Global Head of Discovery Research Coordination at Boehringer Ingelheim, where he is responsible for guiding research policy and strategy. Adrian represents Boehringer Ingelheim on the Board of Trustees of the SGC, and he is also a Vice Chairperson of the Research and Innovation Strategies Group of FPR. Thanks for joining us today, Adrian, and we look forward to your presentation.
Thank you very much for the kind introduction, Anka. I assume you can all see my first slide.
Yes.
Good. Target 2035, a probe for every protein. When we sat together in Berlin in 2018, began to hammer this one out, and also published some work around it last year, a quote sprang to mind from Lewis Carroll, which I've actually used before many, many moons ago. And to give you its full run, Alice laughed. "There is no use trying," she said. "One can't believe impossible things." "I dare say you haven't had much practice," said the Queen. "When I was your age, I always did it for half an hour a day. Why, sometimes I believed as many as 6 impossible things before breakfast." What I'm going to try and do over the course of the next 20 minutes is convince you that maybe Target 2035, a probe for every protein, isn't impossible, and with your help, we'll be able to achieve it. Next, I've got to go through the next slide. Okay, so I think Cheryl has already highlighted the fact that from a pharma's perspective, discovering new medicines is extremely difficult and also increasingly expensive. Much of that high cost is driven by failure, unfortunately. Industry has responded to this challenge by embracing open innovation to access external ideas. Historically, you can see this on the right-hand side of the slide, partnerships were usually bilateral and the drug discovery process was shrouded in secrecy. The model is rapidly changing. With the advent of the internet, drug discovery hopefully has become more decentralized, bottom-up and scalable than ever before. What I'd like to do in the course of the next 20 minutes is walk you through 4 aspects of Target 2035. I'd like to present to you some data which follows on some of the data which Al has showed about too many proteins, too few reliable tools. I'd also like to outline to you some of the first steps we're taking with an IMI2 project called UBOpen, short for Enabling and Unblocking Biology in the Open, which is part of phase 1 of Target 2035. I'd like to also outline some of the steps that we're taking as an organization, Boehringer Ingelheim, in order to be able to make some of our molecules freely available, unencumbered with those associated data packages, so that the broader scientific community can use them without any restriction. And then finally, I'd like to present to you how academia and industry could work together to harness the power of the scientific crowd and achieve the goals of Target 2035. So the first part is too many proteins. We sat down and we used this as a basis for the Target 2035 publication in Drug Discovery Today. We wanted to show the natural tendency for scientists to focus on previously studied genes. You've seen some of this information in another form presented by Aled a few moments ago. What we did here was we used data sets from the NCBI to evaluate the frequency patterns of publications annotated to human protein coding genes. And then we looked at this, we compared this to the online catalog of Mendelian inheritance map, genetic disorders, and we also looked at published GWAS frequency studies. As you can see on the right-hand side of the slide, despite the fact that the number of publications is increasing from year to year, the accumulated number is shown in green. You can see sometimes the backlog that has to be catch up when we did this study, but you can see the number of publications is increasing from year to year. However, when you begin to look on the left-hand side of the slide, and I've focused to you on the lower part, here you can see what looks very much like a power law distribution of the 596,000 publications which were entered into Gene2PubMed, annotated to the different protein coding genes, human ones, in Gene2PubMed. That counts 20,122. What you'll see is a very uneven distribution, similar to that distribution that Aled has presented to you for the kinases. This little fella here on the top left-hand side is the gene that codes P53. I'm not saying this is not an incredibly important tumor suppressor gene. However, it's also one of the most frequently annotated publications in the Gene2PubMed database. If you then look at the top part of the slide, both the OMIM and the GWAS, and remember the OMIM is like a barcode. It just tells you a 0 or a 1, whether there's a link in the online catalog of human genes. And what you see is there is genetic interest across the entire proteome, as you might expect. You can also see the frequency of GWAS hits, but this doesn't seem to influence the way scientists are working on these particular proteins. You still see this imbalance as Al has presented to you before. The next slide. We're not the only ones that have been looking for this, and if you go to the Target 2035 website, you can see a link to 2 of these papers. This is work done by the NIH. They have the US National Institutes of Health has launched Illuminating the Druggable Genome, IDG for short. They launched this in 2014. Their basic assumption was the large proportion of biomedical research and the development of biotherapeutics is focused on a small fraction of the human genome, and they tried to map this. I shan't go into any detail, but just draw your attention to the inner ring of this circle on the left-hand side, which shows you the breakdown into inner ring colors of what they've described as T dark, in which we know little or nothing of these particular genes or proteins, the T bio, where there seems to be some sort of biological inference in there, T chem, chemical, where we know there's potential chemical modulation of these particular proteins, and the T-clin, which is actually the smallest part for which we have clinical evidence that these target classes are relevant. So it's still 3% of the entire genome is what we've probably explored already. Vast amounts of the genome are very underexplored and may actually provide us with some exciting therapeutic opportunities. Next slide. We've coined the term open tools, and I'll go into that a little bit here. We've had a plethora of expressions of open science, open source, open data, open access, different ways of opening up the whole process. But open tools is something which we've coined, which actually encompasses a whole variety of different things. Chemogenomic library members, chemical probes, tool antibodies, bioinformatic and in silico tools, and other agents, all of which are important assets to be used when beginning to interrogate the biology. There's also a slight paradox in here, is that quite often the scientists with very high levels of expertise in a given disease pathway or protein target and thus best positioned to carry out research in these areas, don't always have access to the necessary tool compounds. And this is what we're trying to change with Target 35. What we'd like to do is put these sort of high quality chemical tools and other tools into the hands of people who begin to understand and interrogate areas of biology which are not actually well understood at the moment. The next slide, and it builds a little bit on what Cheryl was saying a few moments ago, is for me a continuum. We work our way from chemogenomic library members, which support the identification of possible target protein families, move through the continuum to chemical probes, which allow us then to interrogate those biological systems in a selective and specific fashion. Well-defined criteria, I'll come to those in a minute. And obviously the goal is, in order to be able to understand the biology, hopefully we'll be able to create the necessary therapeutic or agent which allows us to actually treat those diseases. But each one of those is a tool which is built for its own particular part, and therefore it's always good to understand what the promise and also the limitations of the tool at any particular stage. The next slide shows you perhaps some examples of what you might think of as a chemogenomic compound. Obviously, we can look for different chemotypes, acquiring them, distributing them, quality control, and defining the criteria for these chemogenomic library members. We expect a certain level of general selectivity in sentinel panels. I think the prime example for this is the work which Tim and his team have been doing with respect to the kinase family and the KCGS set. Obviously, if people are going to be using these molecules, these tools in cell systems, we don't want them to be cytotoxic. And we also want to understand what the cellular activity is, not just the in vitro cell-free system activity is. This is in contrast to the chemical probes, and I think, again, Cheryl highlighted the importance here that any chemical probe actually has to fulfill very, very stringent criteria. And if you go back to the article, The Promise and Peril of Chemical Probes, you'll understand that good probes help good science, but bad probes, if you don't understand what the limitations are, can lead to misleading conclusions. And therefore, together with a variety of other experts, we've worked out a general set of criteria which we apply, and also independent scientists in the evaluation process apply to judging whether a probe fulfills the criteria and can be accepted as such. The Target 2035 I like to think of is built on a belief and an assumption and a goal. The belief that we have is they're interrogating the function of a protein, and determining its relevance as a drug target is best done with a pharmacological modulator, be it a chemical probe or an antibody or a chemogenomic library member. The assumption is if these tools are available for most proteins, it should be possible to translate the tremendous advances in genomics into a greater understanding of human health and disease. And finally, our goal is that by developing a global federation, we can develop and apply new technologies with the goal of creating these chemogenomic libraries, chemical probes, and/or tool antibodies for the entire proteome. The first phase of Target 2035 is built around characterizing and distributing existing pharmacological modulators for key representative protein family in the druggable genome and generating some new novel chemical probes for particular family members. We want to develop a centralized infrastructure to facilitate the collection, curation, dissemination, and mining, and also use that to empower the scientific community. We also want to create a centralized facility to provide the quantitative genome-scale biochemical and cell-based assays that will be needed to interrogate that. UB Open has been the first step in this. Open access chemogenomic libraries and chemical probes for the druggable genome, and here you see a cross-section of the 22 different members that we have on board. We started this at the height of the COVID-19 crisis on the 1st of May 2020 without any delay. It has an overall budget of 66 million euros, and the leadership is split between the coordinator in Frankfurt and ourselves at Boehringer Ingelheim. UB Open has the goal of open access chemogenomic library of around about 5,000 well-annotated compounds covering about 1,000 different proteins. We also want to synthesize the next 100 high-quality open access chemical probes. And we've also drawn a mark in the sand. We want to focus on E3 ligase handles, which is one of the classes that Cheryl showed you, I think, in her presentation, and also solute carriers. We want to establish that infrastructure. We want to develop and implement advanced transfer of technologies, disseminate reliable protocols for patient-derived cell assays in human disease tissue, and establish the necessary infrastructure and platforms and governance to seed a global effort. I'll skip over that one. We've already made the first steps in trying to develop a global collaborative network. It's growing by the month, and what I'm trying to do here is depict all the different people and organizations that we're trying to involve thus far in our UB Open initiative. Molecules to order. There's also steps that each individual company, I believe, can make in this whole process. We at Boehringer have developed our own open innovation portal. We call it OpenMe, and their primary aim was to give scientists access to selected, well-characterized molecules from our older projects. We have something called Molecules to Order, which we provide our molecules free of charge without the need to enter into intellectual property discussions. We do clear up certain liability issues, but there's no restriction on intellectual property. We also use it as a platform for making certain molecules for collaboration available, for them to submit research proposals. And we also, in the last few months, have started actually formulating biological questions on what's called our Open to Experts portal. As I said, molecules to order, we provide them free of charge without any intellectual property restriction. We've actually shipped 4,500 compounds in 54 countries over the last 2,500 years, and we've tracked, as if best we can, 40 scientific papers. We don't do that to our exclusion. We also work closely with the colleagues at the SGC and the Donated Chemical Probe Program, which have been outlined to you, to make many of these probes also available there. We actually were very proud a few weeks ago to actually have launched our 50th molecule, GPR40, ligand BI2081. I think this is showing you how far we've come in an organization which didn't used to make such molecules available freely to the general scientific community. As I said, we work together with the SGC and the Donated Chemical Probe Program, which has been outlined to you. And an example of that is a recent potent inhibitor of the human solute carrier, SLC13A5. If you had more time, you could go into a little bit more detail about that one. The final part then for me is how academia and industry can work together to harness the power of that scientific crowd, because I think that's what it's all about. And I always like to think a very neat quote by Thomas Jefferson, that Joy's law means that most of the creative scientific ability is going to be outside of your own organization. He who receives an idea from me receives instruction himself without lessening mine, as he who lights his taper at mine receives light without darkening me. It's all about learning to share. This has been a growing movement, and I actually see Target 2035 launched in 2019, and our UB Open, which is our IMI2 initiative that I've just described to you, as part of that growing movement along the continuum, starting off with close partnerships in the early 80s. 1990s, private companies began to consider public consortium. There are various examples here. The SNP consortium, you see the International HapMap project, the launch of the Structural Genomics Consortium, which you have to give the credit to this organization back in 2004, which actually set this whole process up. The different IMI and IMI2 innovative medicine initiatives, the open malaria source, and I think Target 2035, I hope, will be just as an exciting initiative to be able to participate in. So I hope that I've managed to convince you that a transition to a more formalized federation to avoid duplication of effort and consolidate core activities and share advances more quickly is a sensible thing to do if you want to develop a protein, a molecule to influence every protein in the human proteome. Phase 2 will apply those new technologies and infrastructure developed in phase 1 to generate a complete set of pharmacological modulators for most of the 20-odd thousand human protein coding genes. And we might see alternative approaches. We're already seeing that some very exciting advances with chemically mediated protein-protein interactions, such as proteolysis targeting chimeras, which means that we can have ligands that bind but not necessarily to the active site. You can also influence the function of a particular protein in the body. Which brings me to the last slide. Target 2035, a probe for every protein. We're beginning Target 2035 with the UB Open initiative, but it's only the first step. It's phase 1, enabling and unblocking the biology in the open by building on the success of the SGC. We've also, I hope, been able to show you today that OpenMe is a platform that each organization can think about sharing their molecules, which are probably languishing in drawers, waiting for their sell-by date to actually expire, that there's actually value in using a portal to share those chemical probes. Either do it yourself or come and do that with a donated chemical probe program. I haven't gone into details about negative controls, the whole story, how important it is to have the appropriate negative control when you be able to do the biological experiments. And Al has already highlighted the importance. The probe themselves, their negative controls, are not anything unless you have the experimental data and metadata package that allows you to share that with the scientific community. And I hope that academia and industry can work together to harness the power of the scientific crowd by providing them with the necessary tools for many more proteins than they've been looking at up to now. And this is the goal of Target 2035. And on that note, I'd like to end my talk. Thank you very much for listening today.
Thank you very much, Adrian. And also, again, thank you very much, Cheryl and Alet, for the brilliant presentations. And I think they provide not only a very good overview on what Target 2035 is, but also on the different use cases and the achievements that we have already seen during the past years. We now come to the second part of this webinar, and that is a panel discussion. So please, everyone, if there is a specific question that you would like to see discussed by the panelists, just enter that in the Q&A. It says F&A here on the bottom of the window, and we'll make sure that we find time to discuss at least a few of your questions. And with that, I'd like to introduce the panelists to you. We have, first of all, Derek Lowe. He is a director in chemical biology and therapeutics at the Novartis Institute. Derek is a chemist by training, and he has worked for several major pharmaceutical companies on drug discovery projects. He is an expert on several diseases, including schizophrenia, Alzheimer's, diabetes, and osteoporosis. Our second panelist is Deborah Rothman. She is a biochemist and biologist by training, and in her current position, she is a director of chemical biology within the discovery chemistry at Merck & Co. Her special focus are infectious diseases, cardiovascular, and metabolic diseases. Also on the panel, we have Peter Kirkpatrick, who is the chief editor of Nature Reviews Drug Discovery. He gained his PhD for research on the vancomycin group of antibiotics. Peter is interested in all aspects of the drug discovery process and in facilitating interactions between the as well as between academia and industry, certainly something that Target 2035 clearly needs. Also on the panel, we have Paul Workman, a multidisciplinary cancer research scientist, molecular pharmacologist, and chemical biologist. Paul is particularly well known for the discovery of several clinical drug candidates and for the development of chemical probes for different target families. He is chief executive and president of the Institute of Cancer Research in London and Harrod Professor of Pharmacology and Therapeutics. Paul is also executive director of the Chemical Probes Portal. Last but not least, we have on the panel Susanna Pitancesca, Director for Strategic Development and Partnerships in the Division of Neuroscience at the National Institute on Aging. Her strong focus is on Alzheimer's disease, research and drug development within an open science framework. Susanna is Program Director for the Accelerating Medicines Partnership for Alzheimer's Disease Target Discovery Program and several affiliated open science consortia. So thank you very much to all of you for agreeing to join this panel. I'd like to kick off the discussion with the first question going around the virtual table, and that is, what are your first thoughts to the presentations? Can you please give a short statement each, why target 2035, or if you don't, target 2035? So who wants to start? Maybe we can just do it in the order on the agenda, so that would be you, Derek.
Oh, wonderful. Okay, well, I am very much a huge fan of good chemical probes, but then again, you know, I started off as a small molecule chemist, so I would be, but I think that the arguments advanced for them are completely sound, and I believe that they really do push the field into trying things and into explorations that we have to see. That said, as also mentioned, this is going to be a significant challenge because I have spent my career trying to find useful small molecules that interact with a lot of therapeutically interesting proteins. And in some cases we've succeeded, and in some cases we haven't. There are going to be some classes of proteins that are going to be a really tough challenge. Disordered proteins, transcription factors, others that people have taken some very expensive, lengthy runs at. That said, they are not impossible, but I think what we're going to have is a sort of Pareto's law. We're going to be able to find probes for a good number of proteins, and then there are going to be some very difficult ones to crack, but it's a very worthy effort.
Deborah, would you like to continue?
Sure, thank you. Yeah, I think I think it's a very ambitious goal, and I do think that in the future we will have some technologies and capabilities and creative ways of thinking where we can accelerate this significantly. But I agree with Derek that I think there are going to be some tools or some targets that are very challenging. You know, for example, the cellular context is going to be extremely important in which we try to modulate these targets. And so if we consider really a stripped down protein by protein attack, I think we might miss out on some of the really interesting biology that these targets are producing, whether they need to go from disordered to ordered, or they're in particular complex, or once they're triggered, basically it starts a cascade which can't be stopped. So I think we need to consider all of those biological implications as well while we start to look for these probes. But I do have confidence that in the next 5 to 10 years, we're going to see some capabilities between high throughput chemistry and high throughput biology that are going to allow us to really achieve very close to, if not the whole proteome.
Fantastic, thank you very much. There has been one technical question, maybe I answer that immediately, and that is, how can we ask questions? Please, everyone, use the Q&A. I think the chat function has been disabled for most of the participants, so use the Q&A, or as it says, at least on my screen, it's the F&A, which is German for Fragen und Antworten. So if you want later on to have your question discussed by the panelists, go to the bottom of your screen and click on the F&A or Q&A and then answer your question. So with that, let's continue. Peter, you're next in line. What are your first thoughts on the target 2035?
Well, I think the presentations make the rationale for target 2035 very clear. And in addition, I think not just why it's needed, but perhaps also why now is such a good time for this, an ambitious effort of this nature. I mean, similar ideas have been around for a while, but I think now there are several kind of key components in place, including, what makes a good probe, and particularly, I guess, new tools to develop and model set chemical probes. And I think, with these tools in place, this is much more feasible than it would have been 10 years ago. And I think also crucially, you can see now the kind of enthusiasm and engagement with open science has grown thanks to the efforts of and achievements of organizations like the SGC.
Thank you very much. So Paul, you're next in line. What is your take on the presentations and on the target 2035?
Of course, audaciously ambitious, but I think that's a brilliant thing. It should stimulate the community to get behind this, and the arguments are so well made that I and others have commented as well. What I thought I might do is comment on a question that already came in, because I think, Anke, you also wanted to know what's the connection between Target 35 and your own activities. And so in my activities as a chemical biologist and a cancer researcher, I use chemical probes all the time, but I want more of them. We need them in different target families, and we need high quality, and we need inactive analogs and so on. But the thing I'm particularly passionate about, which is why I, with other colleagues, with Aled and Bistan Al-Azikani and Albert Antolin and Dom Senin-Felice, decided to apply for a grant to take on the chemical probes portal, which had run out of funding really, was because I believe really strongly that it's not just about discovering the probes, but it's finding a way to make the best probes available to biologists, because it's actually the biologists who do both great things and also terrible things with good and bad probes. So a key thing for me is the way I hope to interact with Target 2035, as well as using the probes that come out, is to make sure that they find a way into the chemical probes portal, which then gives the expert opinion and forms a user-friendly interface for biologists to type in their target protein or their chemical compound if that's what they want. And in that way, they can find information that will direct them, instead of going to Google or going to PubMed and finding the oldest and worst compound to use and then publishing a meaningless paper, but rather find the best, most modern, best validated, annotated tool. And so for me, a really crucial thing to consider alongside the really important work which we've talked about already, which is to discover the probes and to make them freely available. It's then to find a way to get to this huge community of biologists who don't understand chemistry and find it difficult to make a choice. And this is not a criticism of biologists. This is, how do we find the best way to outreach to that community? So that's a particular passion for me in relation to this project as well.
Thank you very much. And I think you have touched on something that is incredibly important. It's not just the availability of the probes, but it's also knowing which compound to use or what kind of application, at what concentration, and so on. And maybe we can come back to that in a minute or two. Let's first hear what Susanna wants to say as her first thoughts.
Thank you, Anke. Well, my first impression is that the initiative, while may sound extremely ambitious and downright impossible, that this type of organized and federated global effort is necessary if to arm ourselves, to arm biologists and drug developers with a tool to go and fight the many diseases that we still don't understand well, or that we still need to treat. Otherwise, if we don't have the proper tools, if we, the tools that we have are giving us erroneous signal, or if the tools that are good are not accessible that by those who have the talent and ability to use them, we are really bleeding the knowledge that we need, and we are marching boldly
into drug development with eyes wide shut. And I would, many of the sentiments expressed here, the sentiments actually, the goals expressed in this initiative echo actually the recommendations that our institute, the National Institute on Aging, received related to enabling and accelerating data development for Alzheimer's. So we have, through our several strategic planning meetings, Alzheimer's disease research summits, received a series of recommendations to enable and promote open science in basic research and drug development. And over the last 8 years have been building translational infrastructure programs that have delivered a whole array of novel targets, made them available, but most of them are actually genes for which very little is known. So we ended up with a new bottleneck that was the impetus for us to actually build another set of translational research centers, the Treaty centers, where SGC is one of the leading institutions, and they're tasked with the type of activities that Target 2035 is taking up for the entire genome. So this again, so hopefully this is a proof of concept that this effort can be organized, although in a disease-specific space. And it would be terrific to see this get off the ground so that efforts like the ones that we support and other funders can be plugged in and scaled up.
Excellent, thank you very much. I think it's fantastic from all 5 of you. So that's very comforting because as some of you said, it's a brilliant idea, but I think Paul said it, it's also audaciously ambitious, which I fully subscribe to. Yes, it is a huge endeavor. So there was one question from the audience asking, why was the year 2035 chosen? What's expected to improve in 15 years to make it possible to reach the target? Any one of you who'd like to take that question?
I'm not part, so I'll just chime in. You know, from my perspective, I don't know why 2035 specifically was chosen. So maybe Adrian or others can chime in. But I do think, you know, with the advancement of increasing chemical matter space, right? We have a lot of new technologies that are, I think, within most industries in heavy use, such as Dell technology and mRNA display, where you can, you know, expand your chemical universe. You're going to start to be able to expand your target universe as well. Right? I think when we screen the same old compounds over and over again, we come up with the same targets, even in a phenotypic setting, and that's a limitation. So, you know, that's where my optimism comes, is as we expand our chemical universe, we're going to be able to expand the target space that we can modulate.
I think another point to that is that from a medicinal chemist standpoint, we are past the rule of 5 era. That was always taken a bit too far, and antibiotics and natural products were always this sort of thing off to the side. But we're looking at a lot more diverse set of molecules, larger molecular weight, more complex. Now, it's not that these things don't have challenges, but the sorts of things you see in, say, targeted protein degradation space show you that there's a lot of room out there that we have not been exploring for the past 10 or 20 years that we really need to get into. And we may well have to push into those areas of chemical space in order to realize any kind of objective like this.
Mm-hmm. Thank you very much. So I access to the chemistry that we need to develop the chemical probe. So in your opinion, what really is the biggest step? Is it the chemical space, needing access to new compounds? Is it technology profiling these compounds? Is it resources, mindset, politics, funding, know-how on specific target families? So what is your opinion on that one?
Probably a mix of all of the above. You know,
That's very comprehensive answer.
Coming from having been at Merck now for almost 4 years, I feel like it's. I don't know that chemistry will be the ultimate bottleneck. I feel like we can get through that. The profiling, I think earlier someone had mentioned that the metadata, is extremely important that comes along with these probes. I think when you consider trying to profile, if you wanted to profile, these 20,000 or whatever, 100,000 probes against 20,000 other proteins or functional readouts, that's going to be certainly a limitation. Maybe it's not required, but to a degree, you're going to want some of that counter screening information. That might be a limitation. Really is that translational biology element, right? So we might be able to tickle proteins, and someone asked a question about agonism versus antagonism. I think if we have probes for all those modulations, it'd be in great space. But really taking it from the cellular context to the human patient, I still see as a gap coming from an industry perspective.
Mm-hmm. Thank you. Paul.
Thank you, Anke. I just wanted to come back to the question about why 2035. And
yes,
I just had a quick peek at the paper again. And although it doesn't, on a quick look, it doesn't actually spell out. But what I noticed is that Adrian and the team of authors, first of all, looked back and said how much progress has been made over the last 15 years, I guess, between, say, the genome sequence and 2015, let's say, or from 2005 to 2020. And then they said, going forward, 5 years to another 15 years. So 5 years to coalesce, to bring the community together, to get the resources organized. And then the next 10 years would be the big push so that maybe explains why 15 years before and then 15 years now. And I guess if you make it further away than 2035, it's too far away. It becomes, well, we don't need to be, we've got plenty of time. So it's kind of, although it's not, it's still audacious. It's just about achievable in an asymptotic way. It's 15 years is not that long that you can relax and think it'll all get done by somebody else. And so maybe in a way, I think it's a good period to choose.
Thank you.
I'd add to that. I think we need to have a aspirational and yet a time horizon that's not distant future, but also in reality, the methodology, particular maturation of computational methods, this is rapidly evolving. So this is that inflection point in research where if all the stakeholders agree to organize our efforts differently and point in towards this North Star direction, that actually, and there are enough examples of open science enterprises really being more productive, mm-hmm. and benefiting everybody participating. So I think we have enough examples and proof of concept, a couple of more coming. So it's ambitious, but at the same time, I think we could make this into a realistic goal if we collectively organize around it.
Excellent. Peter, you want to comment on that one or on something else?
Yeah, just related to the question, I guess. But yeah, things that have improved or could improve in the next 15 years. I mean, the choice of 2035, I'm not sure about. But yeah, I think, one area that hasn't really come up so far, and there's a lot of hype in it at the moment, but it's likely to mature in the timeframe of this initiative, is machine learning. And I guess, I think if I was to suggest one thing where this might have an impact, and it relates, I think, to the understanding of, what compounds are doing in cells at a deeper level than just, on the target you're looking at, is really the potential for image-based cellular profiling. And I think, a few companies are doing this already now. And the key thing here, I guess, is that, that they're generating data sets with the purpose of using them for machine learning and not coming back to data sets that have just been generated and attempting to do machine learning on them then. And I guess this is, something that in the next 10 years or so, maybe could really improve the ability to understand what compounds are doing in cells beyond the target you think they're affecting.
Mm-hmm. Thank you very much. We have quite a number of very interesting questions here on the screen. So maybe I start by reading some of them out. So discovering a new chemical probe require a lot of resources and manpower to transform the motivation of the well-funded larger groups to tackle this effort, which already have their scientific safe space to operate. As I see that just younger scientists with more limited resources that are willing to take a risk will not be enough to accomplish the presented aims here. Anyone who wants to take this one, maybe from the perspective of academia, or also from the perspective of publications and collaborations, interactions? Peter, would you like to comment on that one?
This is not the area. I mean, I
know.
I'm doing this. It's interesting to see. My feeling is that you know, as knowledge emerges, a lot of the time that's the key catalyst. You know, so I think it was very clear from the presentation from Alad, you know, that putting a probe out there, you see sooner or later the impact comes out into the community, and ultimately that often translates into the potential to develop new drugs.
Mm-hmm. Paul, would you like to add your thoughts on this one?
I wasn't quite sure. The question was, what's the role of younger investigators who not be so well funded compared to the very big groups? That was that the interaction between the two?
I think there was the notion that the how can you transform the motivation of the well-funded larger groups to tackle this effort, because they already have their scientific safe space to operate. If it's just the younger scientists with more limited resources, it will not be enough to accomplish the aims that are really and truly ambitious.
I see. Okay. I mean, well, first of all, I think there's, you know, this is it's not citizen science, but it's crowdsourced science, isn't it? Everybody has a role to play here. Actually, I think what we're seeing is some of the big, well-funded labs are contributing chemical probes to the community, and there's no doubt that every investigator will benefit. I was just remembering, actually, there was a meeting at Cold Spring Harbor around in the early 2000s after the genome sequence was published, and Mike Stratton in the UK and Matt Meyerson and others at the Broad had suggested, and indeed BRAF was discovered by cancer genome sequencing. And there was a proposal at a cancer meeting in which some of the greats were, you know, I remember Sidney Brenner came, for example. Sidney's been mentioned as a quote for the importance of scientific technology. And the audience was kind of split, actually mostly against, should we sequence cancer genomes at large scale? And when we surveyed the group, a lot of people said we shouldn't do that because, using the US language, we'll all lose our R01 grants if all the money goes into genome sequencing. Then there won't be enough money for investigators to have their own grants. And I remember Sidney Brenner replying and saying, "This will liberate your lab. If you're a junior investigator, you know, you don't have to clone any more cancer genes because it'll all be done for you." And that liberates you to play in biology with the outputs. And I think the same applies here. These chemical probes will liberate the scientific community, young investigators and senior investigators, to address their curiosity-driven questions with chemical tools that will allow them to ask important questions. And I don't think it's the case that it's great if small labs discover some really important tools, but I do see the bigger labs doing it as well. I'm not sure what others think. I think there are several examples of that.
Mm-hmm.
Okay, I think this question also speaks in a way to what are the biggest obstacles and the issue of incentives and the role that funding agencies can play and need to play, and publishers, right? So in our experience, as from a funders or from program development perspective, working in a large funding agency, it is important to have expectations on the program that you're developing, expectations in terms of the goals, expectations regarding whether it is going to be open science and what aspects need to be open, and to really at the front end, when you issue a solicitation for grants, so that those who are competing understand what is expected of them. And so in our experience, what we're seeing, the product, and actually our goal is for all the open science programs that we're building, that their output would lift everyone's boats. And it will, of course, again, the initial battle is in developing these programs and setting the expectations and enforcing the expectations. But once the products start coming, the community participating in developing in these initiatives, and also the external community, they both benefit. Because again, and particularly investigators benefit because, again, there's a lot more data, there's a lot more good quality data, good reagents available with no strings attached, and that is to everyone's benefit. So a lot more talent can be plugged in to these efforts. But it's not a small, this still remains a challenge, because the incentives in academia, the criteria by which one is promoted, when you publish, expectations on transparent reporting, etc. Those are all need to be aligned and pointed into this direction of openness, transparency, and rigor. Otherwise, it will be very difficult to achieve this.
I fully agree, and I think the motivation of the individuals who could potentially contribute is really a factor that is of tremendous importance. So there has been one question. We have now discussed the motivation in academia that goes more in the direction of the motivation of pharma people. So the question is, as far as I understand, pharma usually goes after the biologically validated target, which could be directly brought into the market. How does this kind of on the targets on which nothing is known or published?
Keep in mind that in pharma, we also do phenotypic screening, and we run into molecules that do things that are affecting targets that we do not yet have tracked down or don't really understand. So I can speak from my own experience. I think most companies have molecules in the files that no one was ever quite able to figure out. Either they were from an open phenotypic screening type effort, or they did things in the cells or in the animal models that no one expected and that seemed to be some other effect. And now that might be because we didn't understand enough about the target. That happens. But I think we do have quite a few things in there that are mysterious and worth tracking down.
And from my own perspective, I would also add that pharma is very interested in innovative new targets, because there is so much unmet medical need. If there is a convincing new target, something that can be explored more because we now have the tools in hand, I think that is a tremendous value for pharma also. But Deborah, maybe you want to add to that.
Yeah, absolutely. I guess, one of the questions is what, you have to define your biological validation, right? A lot of the biological validation comes from genetic approaches, which traditionally were just, knockout or knockdown type experiments. And we know that there are many ways to modulate proteins or even non-protein targets that can provide an interesting biology relative to disease. And so I do think that pharma is very interested in those newer targets and biologies, because a lot of the low-hanging fruit has been covered. To to Derek's point, yes, there's a lot of phenotypic screening on green. I think almost pharma now has, and they have dedicated the really interesting novel probes come from, right? You're looking for modulation of particular biology. You screen compounds that maybe have some annotation, maybe not, but you identify, down the road with a lot of work, a novel target, a novel mechanism. I think, that can certainly feed into this target 35. I don't have a creative solution on how to scale that or do it in a systematic way that you'd be able to fill, the whole proteome. But I do think a lot of targets are going to come that way. And I can also say from my industry experience, we have, folks basically scouring the litter probes to be able to bring them in and to try out and see what happens when you test that target in a new biology.
I think you were broken up for a minute there. Can you please repeat your last sentence? Maybe it was just me, but.
Sure, sorry about that. Well, I my experience in industry has been that folks are scouring the literature for first-in-class probes to be able to test them in their biological systems, right? It might not be the best, but you start with the first and you continue to build upon that. And I think that's, for the academicians out there, keep going. We're always looking for the latest and the greatest.
Excellent. We also have now, thank you very much, a number of questions that are going in context. We can be even more ambitious as we are right now. So the first one is, where is it now? Why do we only consider human proteins in target 2035? There are a lot of bacterial, viral, fungal infections that pose significant threat to human health. Aren't those equally important? What is your perspective on that one? And I think, of course, yes, it's true. Is that something we should try to tackle in parallel?
Well, I think we should, but there's the question of resources and how many audacious projects can we, what was the comment, eat before breakfast, or how many impossible projects can we run before breakfast? No, I mean, seriously, obviously these are, we only have to look at the current epidemic to know that viral targets and other, non-human targets are tremendously important. And of course, there are initiatives, there are initiatives to find inhibitors of these targets for example, rare tropical diseases and so on, where pharma doesn't currently go. I think there are such initiatives. I don't think these are mutually exclusive. I think these are probably parallel activities, perhaps with exchange of reagents between, because surely many of the compounds that we find that work on human targets may work on the equivalent, to a degree, on the equivalent target in another organism as well. So there could be synergy between the two approaches.
Mm-hmm. Peter.
Yeah, I mean, I completely agree about the importance of antimicrobial drug discovery. That was my background originally, and 25 years ago, people were still saying there were no antibiotics, and it's still the problem now. But I think one thing that's become very clear, particularly during COVID-19, is that the host response to infection is really important to understand. And I think this project has the potential to really illuminate this, because it's important in sepsis and it's important in COVID-19, and in many other scenarios where you really need to understand what's going on in the host in order to treat the infection properly.
Yeah, I would agree with that. I think one of the other things we've seen from this pandemic is that the host response is important, and also for things like viruses, the viral entry proteins, all human, are very important too. So it would be nice to be able to target all these things along with the human, but I think we're going to make a lot of progress on infectious disease, for one, if we do have a lot more probes just for the human side of it.
Yeah, I'll just add in that, having having worked with some of the infectious diseases research, yeah, I think the pathogen targets are extremely important. They're prioritized for certain patient populations for sure. One challenge is actually identifying, if let's take gram-negative, for example, right? This is a significant challenge. What are those targets for which we're going to be able to get, kind of pan activity and not a lot of resistance over time, right? This is certainly a challenge. And so I think it's going to be a combination of both pathogen targets as well as host mechanisms, again, depending on that patient population, to be able to handle infectious disease going forward.
Yes, fully agree with all these statements. And as you said, the current pandemic taught us all a lot, including, I think, the importance of publishing early and making the outcome of the research being available, as I said, as soon as possible, so that the scientific community can pick up on this. I think this is a lesson that we very clearly learned. So maybe moving on to the next round of questions, what can we do to make the data and the results of our efforts more open so that the scientific community can really pick up on this? Maybe coming back also, Paul, to the probes portal, because that is obviously a very neat tool to
Yes, so I think I think we also have to think, to go back to my earlier point, we have to think about who is the user. And actually, there was a question that appeared and I answered it and it disappeared. So maybe I can just say this quickly because it connects to the question. Somebody asked us, since the ICR is now hosting the chemical probes portal, does it connect to ProbeMiner, which is a resource that Albert Antolin built at ICR, working with myself and Bisan Lazikani and colleagues. And so the ProbeMiner is a large-scale, non-biased approach which connects to Chembl, and where you can type in your target of interest, and where you will get a completely unbiased quantitative assessment, and you can tailor the search so that you can find you can apply different rankings to the search, and that gives you a quantitative analysis. It will rank everything in Chembl against your target, and you can rank it for potency, selectivity, permeability, and all of those things. Then, if you're a biologist, you're then confronted with an output of potentially hundreds of compounds with different activity on your target, which an expert finds useful, but a biologist finds bewildering. So that you can connect from ProbeMiner to the portal, and in the portal, that's where you can find the expert analysis, where 4 reviewers have had a detailed look and provide the data to the user. So I think there'll be different ways of display. First of all, I agree completely with the questioner, which is all of this data should be in the public domain, hosted on websites that are accessible to. And I think all the organizations that have been mentioned in the talks before are doing that. SGC are doing that, OpenMe, Donated Probes, all making the data available. All of those probes will find their way into the portal and be reviewed. That will take time. But I do think we have to once again think about how can we present the information that we as an expert group generate, and how can we make it interpretable and understandable to people who are not chemists, and to biologists and to biologists who don't work with medicinal chemists, but people who just want to get an inhibitor for their target and put it on cells. And it's easy for them to go to Google, and it's easy to go to PubMed. And some of the other resources that we're building, I think, need to be made more user-friendly. And then we need to do outreach and go out and talk at conferences and with social media and with blogs and so on to get the message across to biologists. This is how you can find the best inhibitor or a good inhibitor for your target.
I think it's very important to generate that pull also from the biologists. So I think they should ask what is the best possible tool for this target. And if it's not in the portal yet, how can we find it? Where is it going to be? Why is it not in the portal? Aren't there compounds around? So I think it would be fantastic if we come to that point.
Yeah. What I find when I give talks, so I always include, whenever I give a talk, if it's cancer or whatever, I always include a couple of slides on chemical probes in the portal. And I always get people coming up, and most of them say, I've never heard of, I don't know what chemical probes are. If I want an inhibitor, I go to the vendor catalog. And that usually will give you lots of old compounds, as well as well as lots of new compounds. But actually, quite a lot of biologists don't even know there are any resources at all they'll go to the most cited paper, for example, on a PI3 kinase inhibitor that was published in 1970.
Right.
And use that.
Peter, would you like to add to that? Because, I mean, what can we do? What can you do to make sure that the best possible compounds are used for science and then also in the publications, of course?
It's an excellent question. I mean, I don't work for a primary research journal, but I do
I know
what the research journals that in the company I work for do. And recently we've introduced a sort of substantial number of checklists to among the characteristics that are in these checklists. What are you doing with your compound? What is it, and has it been characterized? And I think as Paul highlighted, this has really been a critical problem. The lack of awareness amongst the people doing the original biological experiments about the importance of these things to the outcome. And as a journal, we're doing whatever we can to raise awareness of it. But I think as a company, other journals are trying to make sure that this is in the thinking of the when they get to the stage of writing up the paper, they're analyzing the nature of the compounds and making sure that the things that should have been checked have been checked.
Mm-hmm. All right, thank you. So we have a number of new questions that have come in the meantime. One of them being, what role do you expect artificial intelligence to play in reaching the target? Do you expect advances in AI to impact the 2035 target, and how do you plan to make the best use of it? Anyone who would like to share the thoughts on this one?
I'll take a crack at it. So I get asked about artificial intelligence a lot. I think AI/machine learning is extremely promising, and I think it is eventually going to be a very large part of our business. But for something like this, where we're essentially going off into the unknown, I think that does not play into its strengths. I think what we can do now is hope for these approaches to sort through knowledge that we already have, find connections that we've missed, and suggest gaps that might be in there at the very most. And that's really getting advanced for our current AI. But to actually try to come up with anything new is really, really a stretch. So I think that's going to be up to us. I think this is going to be human intelligence. Now we're going to generate a massive pile of data, which will be very appropriate if we're curating it right. If we're curating it right, it'll be very appropriate for ML AI approaches to go through, but I really don't expect them to lead the way.
I think what you're saying underlines the need to really collaborate. So there's one question that goes directly in. What is the role of education in seeking to better integrate chemistry, biochemistry, biology to bridge the gap for the accurate implementation of probes? I think this is very important. So how to make sure that the different disciplines work together, and I would really add to that how to make sure that pharma and academia work together. So Susanna, I think you want to say something on this one.
Well, I think this is a critical enabling aspect of this endeavor, and actually collective efforts to have a new generation of translational scientists and drug developers. So from, again, I can speak just from the NIA perspective and what we're doing. We've recognized that the new generation of translational scientists need to have highly cross-disciplinary expertise. So we have training programs where we are asking the academic institutions to develop curricula that are at the intersection of biology, data science, and drug discovery. So that while no one person will be a deep expert on all three, it is really important to be at least conversational and to understand actually all these aspects when it comes to both, on one hand, understanding biology more deeply, and on the other hand, advancing something into therapeutics. So I think training, and so there are different training vehicles, but this is again where funding agencies can really contribute in supporting different ways of training and educating the research community.
Excellent. I think, yes, I fully agree. This is something where funding agencies can be extremely helpful. But I think this is also something that needs to be stressed during university education and maybe even earlier, starting at schools. On this one.
That's right.
Could I add that, I think that the field of, first of all, I agree absolutely with everything that Susanna said. It was a perfect summary of the importance of education and multidisciplinarity and anything that we can do to enhance that. You only have to look at the impact of chemical biology as a discipline. It started out as two different fields. Pharmacologists would argue they've always done chemical biology, but and medicinal chemistry, but the bringing together of chemistry and biology has been in the discipline of chemical biology. And whether it's one individual lab where the PI has both chemistry and biology expertise, or perhaps more commonly where you have a biology collaborator and a chemistry collaborator working together, or teams of people in that way, that shows the power of the discipline. And that's where chemical probes come from, really, from that area. And then similarly in industry, you have medicinal chemists working alongside molecular biologists and cell biologists, disease experts. Again, shows the power of that. I think in academia that you can get pockets of that, but you can also get people who feel very isolated if they're working in a top-class biology institute and there's no chemist anywhere near them. It's much harder for them to find the way forward. And that again, I'll risk repeating myself. That's the group of people we need to find a way to bring into this community, to our community.
Excellent.
The journals do have a role to play, because if the journals get tougher if the journals make sure that they don't publish beautiful papers with fantastic biologically detailed analysis on completely worthless chemical molecules, Michael acceptors, broad spectrum kinase inhibitors, and we see those papers continue to be published. And what it needs, of course, is the journal editors to be educated, and you need to make sure that you have either a chemical biology reviewer or a chemist and a biologist reviewing the paper. If we can increase the rigor of, then people will learn that if you want to get your great biology paper published, you have to use a really good compound as well as doing really great biological experiments.
Right.
I second that. That is so hugely important. I think the journals have a role to play in really raising the bar on expectations on data, method provenance, authentication of reagents, as do funders. But again, there's a lot of room for improvement here. And of course, this is incredibly human intense endeavor. It means that you will require another set of reviewers to adjudicate a paper outside of the merits and the hypothesis and the interpretation of data, etc., on the more dull but critical aspects of what it's actually, what the material, what material was put in to actually conduct it.
Yes. So I think we've talked a lot about the quality that is of utmost importance here, and we have talked a lot about interdisciplinary work. So what kind of additional talent would we need to make Target 2035 a success? There is, of course, the usual disciplines like pharmacology, chemistry, and so on. But how can we make sure that we have enough people to promote, that we have enough people on board who are willing to take a lead in educating? How can we make sure that the initiatives that have been started already around the world are really working together to make sure that we are that we have as little redundancy as possible, that we really agree on how to tackle this ambitious approach, and that we share the vision on quality and also on open publication. Any ideas on that one?
Well, it's a very big question you just asked, and it's probably
Yes.
it's probably the critical one, actually. So the one that's perhaps easiest to answer is, how do we make sure there's no duplication, and how do we make sure everybody benefits? It is just to make sure that the information is all in the public domain in a accessible form that biologists can understand as well as chemists. Then I think maybe we have to learn the lesson from the Genome Project. This is about, and now we see it in proteomics and in other fields. We see groups of people coalescing with a common aim, writing big grants together that can facilitate contributions from multiple partners. I don't think it's quite as easy to put this together as it is perhaps in, say, genetics, where clearly if you want to find a gene or you want to do a genomic study, then the more samples you get and the more investigators all coming together and you pool the data and you have computational analysis. How you build that community, I think, is quite challenging. I actually don't know whether Adrian and Cheryl are still on. Maybe they could comment on how they see it coming together.
Good idea. Cheryl, would you like to jump in and say a few words on this one?
Okay, sorry, could you summarize it again?
Shall I do it? The question was, how do we get everybody to work together, basically? And I was making the comparison of, genomics or genetics, armies of people working together in TCGA and
right.
proteomic, and how do we get people to work together on this initiative?
I think it's going to be a combination of big science and traditional individual lab hypothesis-driven science working together. I mean, as you probably know, the SGC has a extensive track record of working with individual labs. We have some platforms, if you will, for large-scale processing of proteins and testing them and screening them. And our group in Toronto, in fact, has done all our chemistry has been done through collaboration with academic labs, with industry. And so I think in the spirit of, open collaboration, the more we can foster that type of interaction, the more, the network will spread around the world. And I know granting agencies like to see groups work together with complementary expertise, and we write letters all the time for others to get grants and vice versa. I know I think this discussion started with what extra talent is going to be needed as well. And I think engineering can contribute as we need to develop technology and maybe miniaturize some of the activities we do. So bringing in the engineering community is also an important aspect.
Thank you very much, Cheryl.
Could I say one of the things that's also helpful, I think, is the cross-representation. So, for example, Suzanne Muller-Knapp, who's involved in many of these initiatives, is also the head of operations for the Chemical Probes portal. So the sort of and Aled Edwards from SGC is also working with ICR colleagues and so on. So this, I think, cross-representation across the different groups that are all working in this space will help to make sure that everybody knows what everybody else is doing as far as possible.
Exactly. I think that's the goal of building this federation and this community where we can all work together comfortably without worrying that, you know, we're competing against each other, with knowing that we're working together.
Thank you very much, Cheryl. I think these were almost final and brilliantly phrased final words for the session. But before we really close the session, I'd like to thank all panelists, and I like to ask everyone to give a very short final comment. So can be, if you meet someone, how would you in 15 seconds, in 13 seconds, convince them that Target 2035 is a good idea and that they can contribute? Let's do it. Let's start with Susanna this time.
Well, I have to step back from this as I, again, work at the NIH and we're a funding agency. So
I know.
Not to conflict myself here, but I would say all I can say is that our institute, the National Institute on Aging, has a commitment to expanding the open science universe of research and really building these new translational teams that are highly cross-disciplinary to advance the wealth of novel targets already into the drug discovery and drug development more seamlessly. And so it is great to see an effort like this coalescing. I want to congratulate Cheryl and Aled on the ambition to actually to conceive of this and to aim high. And I just look forward to see this community aggregating and so that organizations like the NIH or like the NIA can align in not too much into the distant future.
Thank you very much. And to be very clear about that, when I talk about Target 2035, I do not see a single organization or a single funder in really being the one as a truly global effort and as a federation of different initiatives to hopefully include as many funders, as many labs, as many organizations as possible. And when I talk of this Target 2035, I really see this federation that would include everyone who wants to participate. Thank you. Paul.
Oh, I think everybody said it. So I'm going to answer a slightly different question. You'll see why in a second, which is, how do you convince a biologist to either recognize that this is important or to get involved? And I would say that don't just rely on genetics, that chemical tools are enormously useful. The ability to modulate dose and concentration and time of exposure are tremendously important. It's complementary to CRISPR and RNAi. It will allow you to ask really important questions in your science. And who wouldn't want to have the best available tools available to do their science? And in that way, you know, since we can now do whole genome experiments with CRISPR and so on, similarly, if we had whole genome chemical tools or whole proteome chemical tools, think of the incredible power of that. And who wouldn't want to be part of such a exciting initiative? It's also a great crowd of people. You know, it's a fun crowd of people to work together with a sense of purpose. It's really exciting, actually. I think young scientists, when they see this sort of activity going on, can be really inspired to join us.
Thank you very much. Peter.
I really like Paul's reason, so I'll try and say something. Yeah, I mean, I think I'd like to emphasize, the huge benefits that can come from coordinating activities in an open way, learning from the experience of others, reducing the risk of running down the same dead ends, integrating knowledge from multiple sources to give you more robust conclusions. All of these things are really, the kind of things that an initiative like Target 2035 can do. And I think, it's hugely ambitious, but even if it only made it, to 20% of its goal, that would already be probably quadrupling the number of chemical probes available, and that would have a huge impact.
Thank you very much. Deborah, you're next.
Thank you. Yeah, I think everyone said it quite well. I think the arguments for participating in this collaborative, transparent way is really going to accelerate. I completely agree, Peter. Aim for the stars. If you fall, you land on a cloud, and that's still pretty good, right? Any progress is great. I think as the next generation of multilingual, if you will, interdisciplinary scientists come up, I really do think there are going to be creative solutions towards accelerating towards this goal. And so as a community, can we better parse out the really interesting biology that's going to lead towards better human health? I think that's a really great reason to be a part of this initiative.
Thank you very much. Derek.
Sure. I
Final
guess
comment?
my pitch is very simple. Look at the amount of progress that has been made through really validated chemical probes. Look at the number of biologically important proteins that are out there, and look at how few of them we can actually reach with those probes. Why would you not want to increase that number? We're going to get a lot out of it.
Brilliant. Thank you very much, Derek. And with that, I unfortunately have to close the session. It's getting late. So thank you very much to all the speakers. Thank you very much to the panelists. You really did an outstanding job. I thoroughly enjoyed the session. I hope everyone who listened in also did enjoy this meeting. And don't forget, this is already scheduled for tomorrow. So I very much look forward to seeing all of you there again then. Thank you. Have a nice evening. Have a nice day wherever you are in the world. Bye-bye.
Thank you, Anka.
Thank you, Anka, as well.
Thank you.
Great job.
Thank you.